Hello student, as per you have ongoing about the cells and learning. Yesterday class we have read about the introductions. Actually two classes are already done where we have studied about the introduction of cell signaling, their mechanism, mole interactions, etc. Some basic receptors that has been studied from the two classes. Now, uh, in these classes, I like to teach you about the GPCR. Let's study about this topic. What is GPCR? GPCR is a G-protein coupled receptor that has a broad family, about 800 human genes. Uh, Mostly a common mechanisms we can say. Most in most things it has been using as a work as a receptor, as we have studied in the last classes about the uh, nitrogen oxide, where we have seen this part that um, some signals that activate GPCR and that simultaneously goes on and nitrogen oxide synthesis occur where arginine converts to citrulline and nitrogen oxide that goes to smooth muscles and goes on. So this GPCR actually have a basic um, seven transmembrane membrane that's passed in plasma membrane. It's introduced in seven crisscross systems as shown here. I'm showing you that like this. As you know, it have two part, a small and a large protein, four subtypes, G alpha S, G alpha I, G alpha Q, and G alpha T. Let's study about the structure first. In GPCR, there is a N-terminal that is present outside of the cell membrane, and inside of cell membrane, the C-terminal is present. The seven helicase, that is seven transmembrane helicase. Mostly it is a hydrophobic alpha helicase, and the N-terminals in N-terminals mostly the signals are been bound here, and in C-terminals the G alpha, G beta, and G gamma proteins are present. After the signal binding, they activate and the G alpha get detached from G beta and gamma. G alpha act further. Some cases beta and gamma also act. So here is the mechanism how the GPCR activate. In the seven transmembrane helicase, the G alpha in G alpha, G beta, and G gamma are in GDP form, that is in activate form, or we can say the ground state. When a ligand bind to the N-terminal, then GDP convert into GTP. Convert means it switched to GTP, where the G alpha activates that detached from G beta and G gamma. This G alpha further introduced or activate adenyl cyclase, etc. The GPCR, as I said before, the GPCR have two types, that is monomeric versus heteromeric. Monomeric, only the G proteins is present, G alpha. Sorry, in heteromeric, the G alpha, beta, and gamma is present, the three subtype, subunit. And in small G protein, one single subunit is present. As shown in the table, the heteromeric G protein have seven transferrin receptors, four types, fast sensory neurotransmitter and hormone signaling bind GTP to activate. But in signal in monomeric G protein, it's one subunit is present. It's activate RTK receptor tyrosine kinase that we study in next class or maybe further. Some RAS, RO, RAF, RAN protein activate. It's actually work for long-term cell growth, motility, vesicle transport, same as Heteromeric bind GTP to activate. What is the actual mechanisms of activation of GPCR is that when this ligand bind to the receptor, the GF acts. GF acts guanine nucleotide action factor that switch the GDP to GTP. When the GTP is bind, the G alpha is activated. When G alpha activated, the ongoing works etc. has been done, as you know. Then the main point of G protein is that G protein has its own ability. The own ability is GTPase activity. As we say, gap GTPase activating protein. Here, the gap actually is not a factor or protein that actually exchanges the GTP to GTP, GDP to GDP. Here, the G alpha itself has the mechanism that its actual motive is to GTPase that in GTP form it directly reduced to GTPase. No need of any factors, exchange factor, etc. Its own ability is that. Then four types, four subtypes, uh, that is G alpha S, G alpha I, G alpha Q, and G alpha T. Okay, so next. G alpha S actually increase the amount of adenyl cyclase that solely activates CAMP, thoroughly activate PKA. G alpha A actually inhibit the adenyl cyclase. Due to that, CAMP reduce, suppress PKA. G alpha Q is for phosphodiesterase. No, sorry, not for phosphodiesterase. It's actually activates the secondary messengers that is IP3 and TAG, which used to release calcium and PKC. G alpha T is the phosphodiesterase, which reduces the CGMP to GMP or convert the cyclic GMP to normal GMP, close sodium plus channel. In these cases, the gamma and beta also work. So let's start about GL5S. Directly acts on mechanism. When ligand bind to the receptor, G alpha activate. When it activate the adenyl cyclase. Adenyl cyclase then act on CMP. Then the CMP activate the PKA, protein kinase A, and the cellular effects begin. So mostly this the basic mechanism. There is a factor in G alpha S that is its jam and cause cholera toxin. Here is the mechanism. I'll be left showing you. Novel J alpha cycle, as you know, I have shown you in the last page. In case of cholera toxin hijacking, it's actually lock the G alpha S in the activate state, in GTP state, where due to that reason, it actually uh, continuously activated the system, and due to that adenyl cyclase amount will be increased, CMP level will be increased, PK level will be increased, and goes on. In this case, the activation continue, stimulation continues. This called the cholera toxin. Mostly, here we says the ADP ribosolutions or the specific arginine residues on G alpha S. Okay, that completely destroy the interesting GTPase activities. As I have told you before, that G alpha, sorry, G protein has its own GTPase activity. In this case, it has been destroyed and they can't use the GTPase activity. Then G alpha I mechanism. In G alpha I mechanisms, when basically same as before, but in this case, the ligand activates the G alpha I. G alpha I block or inhibit the adenyl cyclase, the activated adenyl cyclase. Due to that, the CMP level reduce, adenyl cyclase reduced, and pKa reduced or suppressed. And beta and gamma actually act on potassium channel and release potassium channel, the potassium ion. In this G alpha I cases, the whooping cough has been introduced that cause due to its uh, lock systems or hijacking, you can say. The G alpha I actually locked in the inactive form. Due to that, it can't stop the adenyl cyclase or further CMP and PK. Due to that, the adenyl cyclase amount increase, CMP increase. that cause the whooping cough, or we can say pertussis toxin. And this caused in the by ADP resolution of the cysteine residue on G alpha I. Next, G alpha Q. It is actual effect is PLC, phospholipase C beta in the membrane. It actually cleave the PIP2 when ligand activated. G alpha Q activates it cleave the PLC. It activates the PLC. PLC cleave the PIP2 into DAC and IP3. The IP3 act on the endoplasmic reticulum where it's release the calcium, and the DAC activate on PKC. That's all on G alpha Q. Then G alpha T. G alpha T, as I have said before, it is phosphodiesterase break CGMP to GMP when the photon light ray act on GPCR. GPCR activates G alpha T, which act on the PD6 that directly activate the CGMP. Sorry, directly the PD6, the phosphodiesterase, actually um, we can say it's uh, demo, it's act on CGMP and converted to GMP, and uh, that cause the sodium calcium channel inactivate, and due to that hyperpolarization occur. Uh, Hyperpolarization occur due to that. Then at last, it is a basic, most important, we can say, most important signal, epinephrine and glycogen metabolisms. We can say that how fight or flight response occur when the situations come or shown to you. The epinephrine bind to the uh, or adrenaline bind to the GPCR, where G alpha S activate. It's active, then stimulate the adenyl cyclase, CAMP, protein kinase. And this protein kinase A actually acts on and off situation, where in on situation it activates the glycogen phosphorylase, and in off glycogen synthesis inactivate. Due to activation of glycogen phosphorylase, breakdown of stored glycogen into glucose for rapid energy, and inhibition of glycogen synthesis stop the cell from wasting energy on storing the glucose. As we need the glucose for fight and flight, it activates the glycogen phosphorylase. Okay, this is the summary particularly. That's all from today. We'll talk later.